Three-point mapping in detail. Three-point mapping is a genetic analysis technique used to determine the relative order and distance between three genes on a chromosome. It is an extension of two-point mapping, which is used to determine the relative location of two genes on a chromosome. Three-point mapping is important for understanding the mechanisms of inheritance and identifying genetic disorders. Steps involved in three-point mapping: selection of parental lines. Two parental lines are selected that are homozygous for two different alleles at each of the three loci that are being mapped. This means that the individuals in each parental line have identical alleles at each of the three loci. Crossing of the parental gene lines. The two parental lines are crossed to produce an F1 generation of heterozygous individuals that are all identical for the three loci. For example, if the three loci are labeled A, B, and C, then the F1 individuals will be AA, BB, CC, like capital A, small a, capital B, small b, capital C, small c. Then identification of recombinant offspring. The F1 individuals are crossed with each other and the offspring are examined to identify those that have undergone a recombination event. Recombination occurs when the alleles at two or more of the loci are separated and recombined in a new arrangement. Then calculation of recombination frequency. The frequency of recombinant offspring is used to calculate the recombination frequency between each pair of loci. The recombination frequency is the proportion of offspring that have a recombinant genotype. Then determination of gene order and distance. The recombination frequencies are used to determine the order and distance between the three loci. The two loci with the lowest recombination frequency are assumed to be closest together on the chromosome, while the third locus is assumed to be further away. For example, consider three loci labeled A, B, and C. If the recombination frequencies between A, B, and C, A, and C are 10, 20, and 25% respectively, then the gene order is determined to be A, B, C. The distance between A and B is assumed to be shorter than the distance between B and C, since the recombination frequency between A and B is lower than the recombination frequency between B and C. Then three-point mapping is an important tool for understanding the mechanisms of inheritance and identifying genetic disorders. For example, three-point mapping has been used to map the genes involved in cystic fibrosis, a genetic disorder that affects the lungs and digestive system. In this case, three loci were mapped on chromosome 7, and the gene responsible for cystic fibrosis is found to be located near one of these loci. In summary, three-point mapping is a genetic analysis technique used to determine the relative order and distance between three genes on a chromosome. It involves the selection of a parental line, crossing of a parental line, identification of recombinant offspring, calculation of recombination frequency, and determination of gene order and distance. Then three-point mapping is important for understanding the mechanisms of inheritance and identifying genetic disorders. Then Hardy-Weinberg principle is a fundamental concept in population genetics that explains the relationship between allele frequencies and genotype frequencies in a population. It provides a mathematical framework for studying the effects of evolutionary forces such as genetic drift, mutation, gene flow, and natural selection on a population's genetic structure. The following are the major points and steps involved in the Hardy-Weinberg principle, like population assumptions. The Hardy-Weinberg principle is based on 5 assumptions, like the population is large, there is no gene flow, there is no mutation, mating is random, and there is no selection. Allele frequencies. The frequency of each allele in the population can be calculated by dividing the number of copies of the allele by the total number of alleles in the population. For example, if there are 100 individuals in a population and 200 total alleles, and 40 of those alleles are A and 160 are B, then the frequency of allele A is 0.2, like 40 by 200, and the frequency of allele B is 0.8, like 160 by 200. Then genotype frequencies. The frequency of each genotype in the population can be calculated by multiplying the frequency of each allele. For example, if the frequency of allele A is 0.2 and the frequency of allele B is 0.8, then the frequency of the AA genotype is 0.04, like we will be multiplying 0.2 into 0.2. The frequency of the AB genotype is 0.32, while we can get that by just multiplying 2 into 0.2 into 0.8, and the frequency of the BB genotype is 0.64, like multiplying 0.8 into 0.8. Then Hardy-Weinberg equilibrium, the principle states that if the 5 assumptions are met, then the frequencies of alleles and genotypes in the population will remain constant from generation to generation. The equation for this equilibrium is very important, where p square plus 2pq plus q square is equal to 1, where p is the frequency of allele A and q is the frequency of allele B. Then testing for Hardy-Weinberg equilibrium. Deviations from the Hardy-Weinberg equilibrium can be used to infer the presence of evolutionary forces in the population. For example, if the observed genotype frequencies in a population differ significantly from expected genotype frequencies based on the Hardy-Weinberg equilibrium, then this indicates that one or more of the assumptions of the principle are not being met. This can be used to identify evolutionary forces such as genetic drift, mutation, gene flow, and natural selection. The importance of Hardy-Weinberg principle lies in its ability to provide a null hypothesis for population genetic studies. It allows researchers to compare observed genotype frequencies to expected genotype frequencies based on the principle's assumptions and to infer the presence of evolutionary forces in a population. For example, the Hardy-Weinberg principle has been used to study the genetics of diseases such as sickle cell anemia and cystic fibrosis, and to track the spread of genetic traits such as lactose intolerance and skin color across populations. In summary, the Hardy-Weinberg principle is a fundamental concept in population genetics that explains the relationship between allele frequencies and genotype frequencies in a population. It is based on 5 assumptions and provides mathematical framework for studying the effects of evolutionary forces on a population's genetic structure. And the principle is important for understanding the genetics of diseases and genetic traits, and for identifying evolutionary forces in populations. Next, inbreeding. Inbreeding refers to the mating between closely related individuals within a population. When individuals within a population mate with close relatives, it results in a decrease in the frequency of heterozygotes and an increase in the frequency of homozygotes. This leads to an increase in the expression of deleterious recessive alleles and a decrease in genetic diversity within the population. The degree of inbreeding within a population is measured by the coefficient of inbreeding, capital F. F represents the probability that an individual is inherited two identical copies of a gene from a common ancestor. Relatives breeding more than the population as a whole, RB greater than C, is a scenario in which the rate of inbreeding within a population is greater than that expected from random mating. Then major points and examples of inbreeding are B greater than C. Increased expression of recessive alleles. Inbreeding results in an increased expression of the deleterious recessive alleles. These recessive alleles may have been hidden in the heterozygous state in the population, but when individuals mate with close relatives, the probability of inheriting two copies of the same allele increases, which leads to the expression of these recessive alleles. An example, cheetahs have a high level of inbreeding due to their small population size. This has resulted in a high incidence of genetic diseases such as skeletal abnormalities, infertility, and immune system dysfunction. Then decreased genetic diversity. Inbreeding leads to a decrease in genetic diversity within a population. When individuals mate with close relatives, the proportion of genes that are identical by descent increase, which reduces the genetic variability of the population. In case of the northern elephant seal population has been found to have low genetic diversity due to historical overhunting, resulting in a small population size. This has made them more vulnerable to environmental changes and diseases outbreaks. Then loss of adaptability. Inbreeding can lead to a loss of adaptability within a population. A decrease in genetic diversity reduces the pool of available genetic variations that can be selected for in response to environmental changes or new challenges. The Florida panther population has suffered from inbreeding depression due to habitat loss and fragmentation, which led to a decrease in genetic diversity. This has made them more vulnerable to diseases and environmental stresses. Then importance of inbreeding. RB greater than C. Inbreeding can have important consequences for the genetic health and variability of populations. It can lead to expression of deleterious recessive alleles, a decrease in genetic diversity, and a loss of adaptability. It is therefore important to monitor levels of inbreeding within populations and take steps to minimize its impact on genetic health. Additionally, inbreeding can be used as a tool in selective breeding programs to increase the frequency of desirable traits within a population. However, careful management is required to avoid the negative consequences of inbreeding, such as the loss of genetic diversity and the expression of deleterious alleles. Pedigree in detail. A pedigree is a diagram that shows the genetic relationships between members of a family over several generations. It is a graphical representation of the inheritance patterns of traits or diseases within a family. Pedigrees are used to study the inheritance of genetic traits, identify carriers of disease-causing alleles, and predict the probability of a trait being passed on to future generations. Steps in constructing a pedigree. Collect family information. The first step in constructing a pedigree is to gather family information. This includes the number of family members, their sex, their age, their relationship to each other, and any relevant medical history. Identify affected individuals. The next step is to identify individuals who have the trait or disease of interest. Affected individuals are indicated on the pedigree with a shaded symbol. Then determine inheritance pattern. Once affected individuals are identified, the next step is to determine the inheritance pattern of the trait or disease. The inheritance pattern can be autosomal dominant, autosomal recessive, X-linked dominant, or X-linked recessive. Then add unaffected family members. Unaffected individuals are represented by an unshaded symbol on the pedigree. It is important to include as many unaffected individuals as possible to help determine the mode of inheritance. Then indicate carriers. Carriers are individuals who have one copy of the disease-causing allele but do not show any symptoms. They are represented on the pedigree by a half-shaded symbol. Then major points. Autosomal dominant pedigree. In an autosomal dominant pedigree, the trait or disease is caused by a dominant allele and is expressed in individuals who inherit only one copy of the allele. Affected individuals are present in every generation, and there is equally a 50% chance of the trait being passed on to offspring. Like Huntington's disease is an autosomal dominant disorder that affects the nervous system. It is caused by a mutation in the Huntington gene and is characterized by progressive deterioration of cognitive and motor functions. Then autosomal recessive pedigree. In an autosomal recessive pedigree, the trait or disease is caused by a recessive allele and is expressed only in individuals who inherit two copies of the allele. Affected individuals are often born to unaffected parents, and there is a 25% chance of the trait being passed on to offspring. Like cystic fibrosis is an autosomal recessive disorder that affects the respiratory, digestive, and reproductive systems. This caused by a mutation in the cystic fibrosis transmembrane conductance regulator, CFTR gene, and is regulated by thick, sticky mucus that clogs the lungs and other organs. Next, X-linked dominant pedigree. X-linked dominant pedigree. In an X-linked dominant pedigree, the trait or disease is caused by a dominant allele located on the X chromosome. Affected males pass the trait on to all their daughters but none of their sons, while affected females pass the trait on to half of their sons and daughters. Like Rett syndrome is an X-linked dominant disorder that affects brain development in girls. X-linked dominant disorder, Rett syndrome. It is caused by mutations in the MECP2 gene and is characterized by developmental regression, seizures, and loss of motor skills. Then X-linked recessive pedigree. In an X-linked recessive pedigree, the trait or disease is caused by a recessive allele located on the X chromosome. Affected males pass the trait on to all their daughters but none of their sons, while carrier females have a 50% chance to passing the trait on to their sons and daughters. Hemophilia A is an X-linked recessive disorder that affects blood clotting. It is caused by mutations in the F8 gene and is characterized by excessive bleeding and related blood loss. Deviation from Mendelian inheritance and interactions. Next topic, deviations from Mendelian inheritance. Deviation from Mendelian inheritance refers to the inheritance pattern that do not follow the typical patterns of Mendelian genetics. These deviations can be caused by various factors such as gene interactions, incomplete dominance, codominance, epistasis, polygenetic inheritance, and environmental factors. So first, incomplete dominance. Incomplete dominance occurs when neither allele is completely dominant over the other and the phenotype of the heterozygote is an intermediate blend of the two homozygous phenotypes. For example, in snapdragons, red and white flower color alleles are incompletely dominant and their heterozygous offspring have pink flowers. Then codominance. Codominance occurs when both alleles are expressed equally in the heterozygous offspring. For example, in humans, the A and B alleles for the ABO blood group system are codominant, and the AB genotype results in the expression of both A and B antigens on the red blood cells. Next, epistasis. Epistasis occurs when one gene masks the expression of another gene. There are two types of epistasis, recessive epistasis and dominant epistasis. In recessive epistasis, the homozygous recessive genotype at one locus masks the expression of another gene. For example, in Labrador retrievers, the black and brown coat color alleles are determined by the B and E loci respectively. The homozygous recessive small e genotype at the E locus, capital E locus, masks the expression of the B locus, resulting in a yellow coat color. In dominant epistasis, the dominant allele is one locus masks the expression of another one. Epistasis occurs when one gene masks the expression of another gene. There are two types of epistasis, recessive epistasis and dominant epistasis. In recessive epistasis, the homozygous recessive genotype at one loci masks the expression of another gene. For example, in Labrador retrievers, the black and brown coat color alleles are determined by the B and E loci respectively. The homozygous recessive EE genotype at the E locus masks the expression of B locus, resulting in a yellow coat color. In dominant epistasis, the dominant allele at one locus masks the expression of another gene. For example, in summer squash, the green and yellow fruit color alleles are determined by the G and Y loci respectively. The homozygous recessive small y genotype at the capital Y locus is epistasis to the G locus, resulting in a white fruit color. Then polygenic inheritance. Polygenic inheritance occurs when a trait is controlled by multiple genes and the alleles at each gene contribute to the phenotype in an additive manner. For example, skin color in humans is controlled by multiple genes and the alleles at each gene contribute to the shade of skin color in an additive manner. Then environmental factors. Environmental factors such as temperature, nutrition, and exposure to chemicals can also affect gene expression and phenotype. For example, in Himalayan rabbits, the temperature-sensitive expression of the pigment gene results in a darker color on the extremities due to the cooler temperature. In conclusion, the understanding of deviations from Mendelian inheritance is essential for practicing the inheritance patterns of traits accurately. It also helps in explaining the variations in the phenotype of offspring. Then intragenic phage mapping. Intragenic phage mapping is a technique used to map the location of mutations within a gene or a DNA sequence. In this technique, bacteriophages, which are viruses that affect bacteria, are used to introduce mutations into the bacterial genome, and the location of the mutation is then determined by examining the phenotypic changes that occur in the bacteria. So when we just try to learn the steps, we can see there are many steps associated with this technique. First one, preparation of phage stock. The first step in intragenic mapping is the preparation of a phage stock that will be used to introduce mutations into the bacterial genome. This is done by growing the phage on a bacterial host strain and then collecting the phage particles. Then introduction of phage into bacterial cells. The next step is to introduce the phage into bacterial cells either by infection or transduction. The phage then integrates into the bacterial genome and introduces mutations into the target gene. Then screening of phenotypic changes. The third step is to screen for phenotypic changes in the bacteria that result from the mutations introduced by the phage. For example, if the target gene is involved in antibiotic resistance, the bacteria can be screened for changes in their susceptibility to antibiotics. Alternatively, if the target gene is involved in the production of a particular metabolite, the bacteria can be screened for changes in the production of that metabolite. Next, mapping the location of the mutation. Once phenotypic changes have been observed, the next step is to determine the location of the mutation within a target gene. This is typically done by analyzing the DNA sequence of the mutant bacteria and comparing it to the DNA sequence of the wild-type bacteria. Then analysis of mutant phenotypes. The final step is to analyze the mutant phenotype in order to gain insights into the function of the target gene. For example, if the target gene is involved in antibiotic resistance, the mutant bacteria can be used to study the mechanisms of antibiotic resistance. Alternatively, if the target gene is involved in the production of a particular metabolite, the mutant bacteria can be used to study the biosynthesis of that metabolite. This intragenic phage mapping has been used to study a wide range of bacterial genes and has provided important insights into bacterial genetics and molecular biology. For example, intragenic mapping was used to map the location of mutations in the lactose operon of E. coli, which led to the discovery of the lac repressor protein and the development of the concept of gene regulation. Similarly, intragenic mapping has been used to study the genes involved in bacterial virulence, antibiotic resistance, and other important biological processes. Next topic, ordered tetrads. Ordered tetrads are a type of tetrad, a group of 4 spores produced by meiosis in some organisms that are formed in certain fungi such as ascomycetes. They are called ordered because the 4 spores within the tetrad are arranged in a specific pattern, which can be used to determine the genetic makeup of the parent cell. So the first step is meiosis. The first step in the formation of ordered tetrad is meiosis, a type of cell division that produces haploid cells with half the number of chromosomes as the parent cell. In fungi, meiosis occurs within specialized structures called asci or singular ascis. I'm not sure about the pronunciation, so which contain 4 haploid nuclei. Next step, spore formation. After meiosis, each of the 4 haploid nuclei migrate to a different end of the ascus and become surrounded by a cell wall forming a spore. The 4 spores remain attached to each other in a tetrad. Next, analysis of spore arrangement. The spores within the tetrad are arranged in a specific pattern depending on the orientation of the meiotic spindle during meiosis. There are 2 possible arrangements, like parallel or perpendicular. In the parallel arrangement, the 2 spores on one side of the tetrad are of one type and the 2 spores on the other side of the tetrad are of the other type. In the perpendicular arrangement, the 4 spores are arranged in a linear sequence of alternating types. Next, determination of recombination frequency. The pattern of spore arrangement can be used to determine the recombination frequency between 2 genes located on the same chromosome. If the 2 genes are unlinked, like located on different chromosomes, the spores will be arranged in a 2:2 ratio, either parallel or perpendicular. However, if the 2 genes are located in the same chromosome, the spores will be arranged in a different ratio. The recombination frequency can be calculated by counting the number of tetrads in which a crossover event has occurred between the 2 genes. Then, ordered tetrads have been used extensively in the study of genetics, particularly in the analysis of recombination and linkage in fungi. They have also been used to study other biological processes as the formation and maintenance of cell polarity. The analysis of ordered tetrads has provided important insights into the mechanisms of genetic recombination and the behavior of chromosomes during meiosis. Ordered tetrads. Once again, ordered tetrads are a type of tetrad which is a group of 4 haploid cells produced by meiosis in certain organisms, and like regular tetrads, they have spores arranged randomly. Ordered tetrads have spores arranged in a specific pattern that reflects the behavior of chromosomes during meiosis. Importance of ordered tetrad. Ordered tetrads are the important tool in study of genetics, particularly in the analysis of recombination and linkage in fungi. They also have been used to study biological processes such as formation and maintenance of cell polarity. Formation of ordered tetrads. Ordered tetrads are formed through meiosis in certain fungi such as ascomycetes. During meiosis, each cell undergoes 2 rounds of division to produce 4 haploid cells. In the case of ascomycetes, the haploid cells are exposed within a sac-like structure called an ascis. Then spore arrangement in ordered tetrads. The 4 spores in an ordered tetrad are arranged in a specific pattern which depends on the orientation of the meiotic spindle during meiosis. The 2 possible arrangements are parallel and perpendicular. In the parallel arrangement, the 2 spores on one side of the tetrad are of one type and the 2 spores on the other side are of the different type. In the perpendicular arrangement, the spores are arranged in a linear sequence of alternating types. Then analysis of ordered tetrads. The pattern of spore arrangement in an ordered tetrad can be used to determine the recombination frequency between 2 genes located on the same chromosome. If the 2 genes are unlinked, the spores will be arranged in a 2:2 ratio, either parallel or perpendicular. However, if the 2 genes are linked, the spores will be arranged in a different ratio, and the recombination frequency can be calculated by counting the number of tetrads in which a crossover event has occurred between the 2 genes. Next, examples of ordered tetrad analysis. One example of ordered tetrad analysis is the mapping of genes in the fungus Neurospora crassa. By analyzing the spore patterns in ordered tetrads, researchers have able to map the positions of various genes in the fungal chromosomes. Another example is the study of recombination and chromosome behavior in the yeast Saccharomyces cerevisiae. In this case, ordered tetrads were used to investigate the roles of various proteins in meiotic recombination. Next, Lotka-Volterra equation for competition. Graph explanation. The Lotka-Volterra equations describe the dynamics of 2 species that compete for the same resources in an ecosystem. The equation is a mathematical model that demonstrates the interaction between the predator and the prey populations over time. The model is named after the ecologists Alfred Lotka and Vito Volterra, who developed it independently in the early 90s. The equation can be written as like this. We can see where dn1 by dt is equal to r1n1 minus a1n1 n2. And like that, where n1 and n2 are the population sizes of species 1 and species 2 respectively. R1 and R2 are the intrinsic growth rates of the 2 species in the absence of competition. A1 and A2 are the competition coefficients which determine how much the population growth of one species is reduced due to competition with the other species. The Lotka-Volterra equation can be represented graphically as a phase plane diagram with the population sizes of 2 species on the x and y axis. The direction of its shape of the predator-prey cycles in the phase plane diagram depend on the values of these parameters R1, R2, A1 and A2. So one important point to note that the Lotka-Volterra model assumes that the predator and prey populations are the only factors affecting each other's growth rates. In reality, there may be other factors at play, such as environmental conditions or the presence of other species in the ecosystem. For example, let's consider a hypothetical scenario where wolves species 1 and deer species 2 compete for the same food resource in a forest. If the deer population grows too large, they will consume too much of the available food and the wolf population will decline. As the wolf population declines, the deer population will start to grow again due to reduced predation pressure. This creates a cycle of predator-prey dynamics that can be described by the Lotka-Volterra equations. The Lotka-Volterra equations are important because they provide a framework for understanding how 2 species interact with each other in an ecosystem. They can help ecologists predict how changes in the population size of one species will affect the population size of another species and how the 2 species will coexist over time. The model has also been used to investigate the effects of human activities such as hunting or habitat destruction or predator-prey relationships in natural ecosystems. Thank you for listening. Next topic we will be learning tomorrow. Thank you.